you, Beth. Thank you, Amy, and thank you all. And thank you for some of the people who have already come up to me and know me from either my work or from students I've had in my class. And so I'm going to really try not to give away any spoilers. That is my goal, okay? And I have my friend Alyssa here who is going to keep me, yes, no spoilers. So before I introduce the movie, I actually saw this movie in 1997 when it came out. I was a child, of course, and it's about 30 years ago, right? I want to admit that having seen this movie throughout the years, and given that my work parallels some of the actual main theme of the movie, which is genetics and DNA, my perspective has changed widely. And from the very first, and I actually saw it recently just to prep for this science talk. And I don't want to get into too many of the details, but I want to introduce the movie. So here in this graphic, the story focuses on these three main characters. Essentially, that DNA helix is the main theme. The idea is that DNA, perfect DNA, is the ideal. And that's what we all want to achieve. And the two characters, the gentlemen here, I'll go by there, Jude Law and Ethan Hawke, sort of have a relationship that works for both of them, so that Ethan Hawke here can achieve his wishes and work in the space program. And so the movie talks about the valids and invalids. Essentially, that's based on your genetic makeup. And the ideal, or the perfection, is laboratory engineered babies, where you design your own baby a la carte. You can, it's also a movie about statistics, and I'm not going to go too much into the details, but essentially removing the risks of disease, removing this, etc. But there is this character of Ethan Hawke who is conceived the old-fashioned way, and he's deemed an invalid. And so the theme of the movie centers along this concept of the invalid versus the valid, which are the ideal in this society, in this world. And so when I was researching this talk for this science talk, I was so amazed, because I don't remember the ad in 1997, but the tagline for this movie as a means to advertise it was that at GATTACA, it is possible to engineer your offspring, right? And this was an ad, it was a gimmick, but what's shocking, and I have it in bold, is that 50,000 people called in, right, interested. And so how can you watch this movie and not think of eugenics, right? I would be remiss if I didn't talk about eugenics. So eugenics simply is a word derived from the Greek, and it is defined as, right here, the practice or advocacy of controlled selective breeding of human populations, as by sterilization, to improve the population's genetic composition. Eugenics was coined by Francis Galton, and Francis Galton is, if anybody here who's ever taken my class, is the cousin to Charles Darwin. Yes. And he coined the term in 1983, and essentially the goal is to make perfect people through genetics, to eliminate, they call the financial burden on society posed by the unfit. And of course, this is an immoral and a pseudoscientific theory, without question. So Galton, in his book, essentially outlines the prescription, what you need, but essentially his thesis comes down to the man is gifted with pity and other kindly feelings. He has also the power of preventing many kinds of suffering, okay? So I conceive it to fall well within his province to replace natural selection by other processes that are more merciful and not less effective. This is the aim of eugenics. And so what is shocking to me was that for all this talk about eugenics, science and society at the turn of the century had no understanding of genes and genetics whatsoever. So the idea of a gene, this physical entity, was proposed in 1917 and still controversial in the 1930s. This idea is DNA the genetic, the heritable material, or a protein? Very huge discussions. The fact that the genes were made of DNA was discovered in 1944. The structure of DNA, elucidated by Watson and Crick in 1953. The code was deciphered in 1961. And of course, Sanger sequencing, where you can actually, base by base, read each letter, was 1977. And importantly, the human genome, which I'll talk about very briefly, was partially sequenced in 2003, but really completed only recently. So I have to give these hidden assumptions behind this simplistic view of eugenics. Bear with me, please. So what are the main assumptions? The first assumption is that genes are rigidly determine phenotypes. Phenotypes is what you see. So if you have blue eyes, that's your phenotype. Genotype is what you don't see. That's in your cell. So false. Epigenetics regulates the expression of genes. And in the last 30 years, we've learned a lot about epigenetics. Epigenetics is driven by nutrients, stress, contaminants, infections, etc., non-genetic, essentially. So genomes are flexible in their response to environmental stressors, or life would never have survived. A second assumption, one gene, one phenotype. False. Most phenotypes, again, what you see, are influenced by many genes operating in concert. Hence, polygenic risk scores, which I'll briefly talk about because this is sort of the work we do in my lab. And a third main assumption is that a genotype is adaptive, meaning favored, in one environment will be superior to other genotypes in another environment. Absolutely false. Natural selection works in response to environmental selective pressure. A genotype that is adaptive in one context is maladaptive in another. I wanted to bring up this example, and maybe people in the audience have read about it. This was this experiment that was done in China on two girls where the scientists actually used a CRISPR technique, which is a gene editing technique, to knock out this variation in an immune cell, okay? And so he thought that by doing this, it would protect against HIV infection. And so what happens is, yes, if you have the CCR5 deficiency, it will protect from HIV infection, that's adaptive. However, the absence of HIV, but you have this loss of function, shortens the lifespan due to impaired immunity. So you fix one thing and you break another, essentially. So these were the hidden assumptions behind simplistic eugenics. And so I want to just read this one quote that Ethan Hawke's character, Vincent, says, "I belonged to a new underclass, no longer determined by social status or the color of your skin. No, we now have discrimination down to a science." And so when I rewatched this movie, it occurred to me, like, what was the inspiration for this movie? What was happening at the time? Something very important was happening at the time. The Human Genome Project. The whole idea of the Human Genome Project is to map and sequence the whole genome to basically classify all the genes in the human genome. This began in 1990, was a huge international effort, and the regions of the genome, the part that's considered important for proteins is only 3% of the genome, and the other large percent, 97%, is what used to be called junk, and I think people call stuff junk when they don't really know what it does, because it's certainly not junk. And there was this race to the human genome. Of course, this is all the science gossip. There was Celera, et cetera, just to say that the working draft was completed in 2000. And then in 2003, there was a publication. And I believe it was Clinton and Tony Blair who said that it's the dawn of a new day, et cetera, in this. about the human genome being completed, but it wasn't really complete, but the public didn't know that, but it was complete enough, so to speak. And so what are the goals of the Human Genome Project? And briefly, it's really, the main goal was to determine the DNA sequence of the human genome, because the idea was that if we understand how the genes work, then we can fix it, and we can, you know, make medicine, help people, public health, medicine, treatment, etc. It was also there to have all this data freely accessible in public databases, which is because it was done with taxpayer money. So it's, of course it's free. And then this idea of human genetic variation, which is we're all 3 billion bases of DNA, but there is variation among all of us. So how important is that variation, especially with respect to disease? We need to find out. Facilitate comparative genomics, yes. You know, what happens in a mouse biologically may also happen in a human, so let's figure out the sequences of other species. And then, of course, this huge drive in technology and the development of sequencing, genotyping, functional genomics, et cetera, and also bolded here, this idea wasn't lost on them, that we have to understand the implications, the ethical, the legal, and social. And so here's, you know, I can't do science without a timeline. And so it orients me as well. And so here, 1992, the human genome, and all of the discoveries throughout, and especially with respect to technology, the dawn of personal genomics, which I'll talk about briefly, and all these initiatives that came out of the Human Genome Project. And of course, recently, the telomere to telomere, so parts, there's repetitive bits of DNA that's very difficult to sequence across, and so that's part of the reason why it wasn't finished. But technology advanced, you know, and of course, eventually they were able to really call each letter of the DNA across all the chromosomes, and it was recently finished, and they called it telomere to telomere, so chromosomes have these ends, and so they were able to finally do that. So why am I here? Well, I'm here because I was asked to be here, and I love the movie, but also because my work actually is a product of the Human Genome Project. I study DNA, I study genetics. I don't study Mendelian diseases, but I study genetics with respect to cancer, as Beth said. And so in this one slide here, this is how I use the information, okay? So I've used the information from the Genome Project with respect to the variation that exists in the Genome Project. I wanted to understand if this variation, is there a differential between diseased and non-diseased? Here, controls or cases. And the lab uses these platforms, these little glass slides that has all this information. And the work that we do where we have genetic variation in very large populations, the results are statistical. And so in order to we have all the statistical data, and we have this beautiful characteristic plot we call the Manhattan plot to express the differences, statistical differences across all this variation across all the chromosomes. And so in my lab, I was the first, of course, with a large group, because we're nothing without our group, to publish on the genetic predictors of endometrial cancer, which is the cancer that I focus on. This was the very first genetic study on this particular disease, and it's in the consortium that I lead. And so then we followed up, and we looked at multi-ethnic populations, and then we followed up again into a larger study. So the point of these 3 papers is to show that it's an ever-evolving process, because there are differences across populations. The more you look, the more you find. And so this was the first step in doing a lot of the work that we do in my lab, which is to understand the role of genetics and environment, this interplay between genes and environment. Because for the most part, for common cancers, which affects 95 to 98% of the population, they're not really described solely by mutations in genes, but rather this combination of genetics and non-genetics. And so in this consortium that I lead, some of the papers that we worked on, we use the genetic predictors, plus lifestyle data, plus clinical data, plus all the data to understand risk factors for the disease. And so these are some of the papers that we've published. In the, the red is obviously the bad risk factors, and then the good risk factors. And so how do you synthesize all this? So first and foremost, we want to improve biological understanding by using the genetic and the non-genetic. And then we want to use to guide education for a healthy lifestyle. And we also, one of the goals is to put together, develop these screening tests to identify high-risk women. This particular disease is a disease that affects only the women because it's a uterine. How do we, is there a way, because prevention is your best friend, right, for cancer, to prevent the disease before. And so we use genetic data, we use lifestyle data, we use all the data that we have in our arsenal to create these statistical methods, like a polygenic risk score. And again, when you watch the movie, you realize, without giving away any spoilers, that it's heavy statistical. The odds of having this, the risk of having that. And one of the great initiatives that came out of the Human Genome Project is the Precision Medicine Initiative that President Obama introduced. And essentially all it is that we know people are unique, and so rather than using one-size-fits-all way to treat people or do medicine, so to speak, we should figure out a way to make it more individual or more personalized, so more targeted. And one of the examples Obama makes in his opening when he talks about this is the same way when you use blood type to match blood transfusion, right? You want to be able to. But from this precision medicine initiative, we have had a lot of progress, a lot of opportunities, as well as pitfalls. And so the way the National Cancer Institute, which is the institute that funds all my grants, which is one of the institutes of the National Institutes of Health, and this is specifically for cancer. It's a form of medicine that uses information about a person's genes, proteins, and environment to prevent, diagnose, and treat disease. In cancer, personalized medicine uses specific information about a person's tumor to help diagnosis, plan treatment, and find out how it's working, and prognosis. And this is my bias, but we have made huge strides with respect to not just to cancer treatment, there's specific treatments for breast cancer with Herceptin. So if you know there's a genetic mutation in this cancer, it gives you a more, if there is a treatment, a more precise way to treat, as opposed to, carpet bombing an individual with just any kind of chemotherapy or indiscriminate treatment that there is. And so the Precision Medicine Initiative or research agenda has made, again, incredible strides. So CRISPR is a gene editing technique. Again, it cuts the DNA and you're able to fix stuff, and it's being used for sickle cell anemia. Here's where I know the best information, and we have great results for targeted cancer therapies. So if you know there's a mutation you can target and there's a treatment that targets that mutation, it's much better, again, than using these broad treatments. Polygenic risk scores, we develop those. Anybody who has genetic information as well as other risk factor information can develop these powerful statistical tools that can predict. We're not there yet, but we're getting better with the prediction. And then, of course, pharmacogenomics, which was one of the very first reasons for wanting to understand variation and genetics because of dosing, right? To get the proper dose of a drug, so that this old method of, let me give you something, and if you don't die overnight, then it was the right dose for now, So just a little bit more precision, so to speak. So, but there are still puzzles, as I say, in the precision medicine world, and of course, improving equity and access, right, to precision medicine. I don't want to read off the slide, but there's what really struck me when I read this article in the Harvard Medical Journal, the Harvard Medical School magazine, was it's more of a philosophical discussion, which is the new normal. And it's not philosophical in my world of telomere research that I do, because whenever we do telomere work, people say, well, how would you operationalize that? And I say, well, there's really no normal because it's so individual. And so where are we? We're again, no spoiler alerts, but we are well into having these tests available to everyone, consumers, right? These direct-to-consumer kind of tests where you can measure your telomeres to see how long you're going to live. Don't get me started on the challenges and pitfalls. Again, there's a lot of assumptions that are made with these tests, and that is this particular test here, which is that there's a knowledge of risk and statistics, and it's not always the case. So this is the 23andMe, it's fun to know, party conversation about your ancestry, but they also gave risks of developing other kinds of diseases. There's a long history with 23andMe. They were taken off the shelf, they were put back on the shelf because and I get it, the founders, they wanted to empower people so that if you know your genetic information, you can have some control. So that is, slippery slope, because it is not one gene, one phenotype, right? So there's a little more nuance to it. So anyway, we've come a long way with the aid of the Human Genome Project, but this is more editorial. Some of the promises of the Human Genome Project were still not there yet. And some of it was a little bit optimistic too. Anyway, I like to end on an optimistic note. That's my story, and I'm sticking to it. And what are some of the benefits? Of course, you want to be proactive about your health. You want to prevent, because it's much easier to prevent than it is to cure or to treat. And personalized health recommendations, because we are unique. We have different lifestyles, we have different genetics. And of course, for me, also understanding etiology, research, and development. And my last slide is the most important slide, which is my group, because without them, none of this could ever be done. And with that, I want you to enjoy this great, great movie.